Okay, so this is my second time attempting this. I hope it goes better than the first time because that was nearly 30 minutes long. Way too long. I shouldn't have done that. Anyways, so let's talk about homeostasis. Homeostasis is the body's ability to maintain a stable environment, internal environment, compared to its external environment. So what this means is that maybe when it's hot outside, you want to cool down, right? You don't want to be uncomfortable and hot. So your body starts to heat up, but it doesn't like that. And you want to try to go back down to a normal temperature. And the way you would do that is you would sweat. If you're hydrated, I have to restart this because it's bad. Okay, this is me restart. Okay, so homeostasis. Homeostasis is the body's ability to maintain a stable internal environment compared to its external environment. So say that it's really hot outside. You're uncomfortable, right? You feel hot, and the only way to kind of help your body cool down essentially is to sweat. Your sweat glands will produce sweat in order to cool yourself off. And the reason that your body does this is because you don't want to overheat. You want to be at a set point, and your set point is going to be referred as the constant or the normal range of which your body has or wants to be at. For example, your body core temperature is 98.6 degrees Fahrenheit. And with this, you can see that, sometimes our temperature is affected differently each day. Like when you're going up the stairs maybe to class, you're getting really warm, but then you come back, So, for example, maybe you are walking to class and you're going up the stairs and you get really hot, but then you finally get to class and you get to sit down for a while and it's really nice, but then you have to walk all the way across campus for a little bit, and then you finally get to sit again and you're good. It's not gonna look like that, but that's just what I was thinking. So we have our body core, which is 98.6 degrees Fahrenheit. That would be different for each person, especially in different areas of where people would live. I would assume that would be different. But yes, this would be what your body is wanting to stay at. You want to be at a comfortable temperature for your body. And the way that the body does this is by detecting change through a receptor. And then the receptor sends a signal over to the stimuli, which is what causes change in, what was I saying? Not volume, variable, that causes the change in variable. And then it communicates to the control center, and most oftentimes the control center would be the brain, because the brain releases hormones and different chemicals to maintain the body's stability or homeostasis. And then the control center will send a signal and communicate to the effector. So let's say that we're touching something really hot. Your brain, your body, the skin, the receptors on your skin are going to detect the thing that's hot, and then the receptors are going to communicate to the stimuli to be like, we need to do something to change this because this isn't right, this doesn't feel right, this is going to hurt us. So then it talks to the brain, and the brain is the executor, and it'll execute the function to the effector to remove your hand from the stimulus, so that way you don't burn yourself. And this is called the afferent pathway. When you're going from the stimuli to the control center, and when you're going from the control center to the effector, that is known as the efferent pathway. And with these set points and everything like that, sometimes you'll stray away from it, or you'll try to go back to it. And that would also be referred to as your negative feedback. So negative feedback is when you're trying to get back to your set point after being strayed away from it. The goal is to maintain stability. You want to be in a homeostatic state, if that's a word. And an example of that would be when you're cold. Like here in Rexburg, it gets cold, right? So when you're cold, you can put on a jacket, but sometimes that just doesn't work, and that's okay. Your body has different ways of being able to warm yourself up. Like right now I'm freezing. I could be shivering right now. That's not something you necessarily control. It's an involuntary control that the brain just stimulates the skeletal muscles to vibrate really fast in order to generate heat, and that way you're able to retain it as well. You can increase metabolism. That's a way to heat up the body as well. Like when you're running, you're increasing your metabolism, the way your body breaks down cells. And then we also have positive feedback. So positive feedback, with this one, you're going away from the set point. And this one is deemed as the one that's kind of like bad. Like you don't want this to happen. An example of positive feedback would be childbirth. So when a woman is giving birth, the baby is pushing on the cervix, and the motion of pushing on the cervix is releasing a hormone called oxytocin, and oxytocin is going to increase the strength of her contractions. That way she can push the baby out. And I think that's really cool. Or so basically the goal of positive feedback is to amplify the response until the outcome is achieved. And with the baby situation, your body's going through so many different states of uncomfortableness. That's not a word, doesn't matter anymore. In order to get this baby out, because at this point the baby's been in there for so long. Another example of this I could maybe think of is a gunshot wound. When you have a wound or something like that and it's actively or profusely bleeding, we want to stop the blood, right? But if you don't have anything to be able to stop the blood, or you're like unable to, the body's going to notice that, hey, this area is losing blood. We should pump more blood over here because it's losing blood. Well, that doesn't work, because now you're losing even more blood because the body's trying to pump it to an area that's losing it. So it's just taking you away from your set point. That's what's happening, and this is homeostasis. Okay. So now we're going to talk about facilitated diffusion, and what facilitated diffusion is, it's different than simple diffusion. With simple diffusion, you're going from a concentration of high to low. And with facilitated diffusion, you're moving substrates, you're moving substrates, I don't even remember now, substances across the membrane that are either too big or they're too polar. And facilitated diffusion, they're integrated by integral proteins or carrier proteins. And this type of diffusion doesn't require any type of energy. And since they're either too big or too polar, they need help getting into the cell. And this is the way that they get into the cell, is through these channel proteins, which are ion specific, which means that they can't just let in any ion. They have to be specific for the cell to let it in. And it moves solutes down their concentration gradient, means it's going against the concentration gradient. And something that's different about this type of facilitated diffusion and channel proteins is that channel proteins open on both sides. So if something wants to get in, it can. If something wants to leave, it can. So we have different types of channels. Someone's yelling. We have voltage gated channels, and with voltage gated channels, there needs to be a change in voltage that binds to this receptor. And then this receptor, so it's originally closed, right? But it's going to open now that it has binded to, say, let's see, calcium. Calcium has a charge of +2. Calcium is going to open this, and it's going to be able to go in. And it is very sensitive to change in voltage because of the amino acids that make up these protein channels. And then you have ligand-gated channels, and an example of this, so they have to be on the extracellular surface. And an example of this would be acetylcholine binding to a ligand-gated channel. So originally, this channel is closed, right? But acetylcholine binds to the ligand-gated channel on the skeletal muscle, and then that allows it to go in. Mechanically gated. These ones, my gosh, why did I say that? Kill me, please. Okay, mechanically gated ones, those ones only open because of change in pressure, touch, or even temperature. So those ones are gonna open and let the ions through because there was a change. I don't know if pH would change it, probably not. I don't know, I'm not gonna get into that. But leak channels, something interesting about these, they are open, always open. So a great example of this would be potassium. Potassium is always going in and out of the cell, and this contributes to the electrical potential in the membrane. That's something we can get into later. But now we're gonna go ahead and talk about the other part of facilitated fusion, which is carrier proteins. And the thing that's different about carrier proteins compared to channel proteins is that these don't open on both sides. They only open on one side. And when they do open, they change in shape. And the difference between this as well is that they can become fully saturated if the concentration in the cell is high enough. Okay. Active transport. It's a different type of diffusion. So this one is different from facilitated in the sense that we actually need energy in order to move things across the membrane or into the cell. So it requires energy in the form of ATP, which is very common within the body and seen almost everywhere. And there are two different types of active transport. You have primary active transport, and that one's gonna move ions against their gradient, and this one uses carrier proteins. So this one also uses ATP directly. Uses ATP, and let's see. So we have something called the sodium-potassium ATPase pump. And sodium is always going to want to leave the cell, while potassium wants to come in. And with the sodium-potassium ATPase pump, you're pumping out 3 sodiums for 2 potassiums, and the only way that this can happen is through an ATPase pump. So these ions are only able to move because of ATP. The ATP is helping them, it's giving energy in order for these ions to move. But with secondary active transport, it also moves ions against their gradient, so out, and then it uses an indirect use of ATP. It doesn't use it to move cells, it uses it to store within the cells. And it has 2 different types of ports in it. You have a sim port, sim meaning going the same way, so it's it's going down its concentration gradient. And while it's going out of the cell with its gradient, against its gradient, it's also pulling in another solution, but with the antiport, anti meaning against, this one pushes solutes out of the cell instead of bringing something in with it. Okay, hopefully that stays once I move. Okay, so now we're going to be talking about osmosis, and osmosis is going to be the movement of water in or out of the cell, depending on the solute concentration. Someone's walking by. And the only way that this can occur is through selective permeable membranes, like channels. That's not channels. I don't know what I'm talking about anymore. But it goes from a low concentration to a high concentration, and a key factor of osmosis as well is that it's permeable to water. And with water, there's the saying, water will follow wherever the party goes. And so it's going to migrate to wherever the higher concentration is. It's going to want to go to wherever the party is, where there's more people. And then it's impermeable to solutes, so it can't let solutes in. Hence the selective permeability. And then it's also passive diffusion because it doesn't require the use of ATP whatsoever. And then we also get into the talk of osmolarity. Osmolarity is a number of moles of particles per liter of solutions. And with osmolarity, you can't see it because I don't know why I wrote it right there. Not osmolarity, tonicity. We're talking about tonicity now. It's how the solution is going to affect the cell and whether or not it changes the volume of it. It's impermeable to ions. Impermeable ion use. Okay, let me rephrase this. Impermeable ions end up using aquaporin channels in order to leave. And aquaporin channels are water-based. So let's see. Yeah, aquaporins are water, and that's how ions leave through these channels in order to reach a state of equilibrium. And with tonicity, you end up going from a concentration, of high to low. So, because water's leaving the cell now. We have different types of solutions. So with our different types of solutions, I can't bend down, it hurts. We have isomotic, and this is referring to the inside of the cell. So we like to say that 300 milliosmoles are the perfect state in which the cell likes to be. And then the same for tonicity. Tonicity, isotonic, it's also going to have 300 milliosmoles. And then with a hypertonic solution, hyper refers to an excessive amount or the overuse. So with hyperosmotic, that means we're going to have more in the cell than we will outside the cell. So let's just pretend that everything basically moved inside the cell and there's nothing left in there. You would say that this is a hyperosmotic cell and a hypotonic solution because there wasn't anything else in there. And we can also get into where these numbers come from, because they do come from somewhere. So you have saline, so you have a 0.9% solution of saline, which is equal to 300 milliosmoles, and that can refer to either here or here. And then you also have a 5% dextrose, but the thing about dextrose is that it dissolves. It's glucose. It dissolves. So once it goes into the cell, it just dissolves. So that means you're going to have less tonicity and the same amount because it's dissolving, so it's not changing anything in here. But 5% dextrose is 300 milliosmoles, and if you were to, let's say, combine the two, that would equal to 600 milliosmoles, and this would be a hypertonic or hyperosmotic solution, or hypertonic depending on where it is. So hyperosmotic We have more in here than we have out here. And if we have more out here, it would be hypertonic. And with a hypoosmotic cell, we end up having less in here and more out here, which would be if we had a hyperosmotic cell, we would have a hypertonic solution because there's more in there. And then because water's leaving the cell, that's the thing. If water's leaving the cell, like right here, if it's leaving, let's show that. So we have, let's say this is our water droplet and it's leaving the cell. That means the volume of the cell is going down, which would put it in a hypoosmotic state, but we're contributing more into the tonic solution, so that would put it into a hypertonic solution.